Hello and welcome you all to the week 6 live session of experimental biochemistry. Hello and welcome to the week 6 live session of experimental biochemistry. I am Surasree. I am a Prime Minister's Research Fellow working in the Department of Biotechnology, IIT Kharagpur. So let's begin today's session. In today's session, we will be covering these topics: polyacrylamide gel electrophoresis and agarose gel electrophoresis. Now, what is gel electrophoresis? So basically, what we have is a protein sample. Either it can be a protein sample or it can be a DNA sample. Now, we want to run this protein or DNA sample in a gel. Now, what is a gel? A gel is a matrix. A matrix, which is actually a polymer. So it is porous. So it is also inert. It is a chemically stable matrix, and it has a particular pore size. Now, when we want to run this sample, run our protein or DNA sample on a polyacrylamide gel or an agarose gel, then what we have to do is we actually apply an electric field. So our molecules, either it be protein or DNA, they migrate along the electric field. Now, if that has to be done, then our molecules have to be charged. So the DNA, RNA, these nucleic acids, when we run on the gel, they are negatively charged. But the proteins can be negatively charged or positively charged, or they can be neutral as well. So what we have to do is we additionally have to charge our proteins, and what we usually do it with sodium dodecyl sulfate, which is SDS. These things we are going to discuss later on. But when this charged molecule, so suppose this is the charged protein or DNA molecule, when this charged molecule will be under a constant electric field, then it will try to move in this gel matrix with a particular velocity, which is called v here. So the electric field is E, velocity is v, but this molecule, it has a particular size and a particular shape and mass, because of its mass and because of its shape, it will also go through a viscous drag. So it will also suffer a viscous drag. So here the velocity of the molecule, of this charged molecule, can be given by v equals to E into Q by F. So which means that the velocity is dependent on the electric field, the charge, and the frictional coefficient, which is the F. So we know that in a gel electrophoresis, we are applying a constant electric field. So therefore, the electric field is constant. So in that case, the velocity will be depending on Q by F. So which is actually the charge divided by the frictional coefficient. So this parameter here will depend on the mass and the shape of the protein or the nucleic acid, and also the charge of the nucleic acid. Now this is the principal theory of gel electrophoresis. Now coming to polyacrylamide gel electrophoresis. So polyacrylamide gel is composed of two molecules, that is acrylamide and bisacrylamide. So this is acrylamide and this is bisacrylamide. These are two molecules. So what happens? Acrylamide, this has this double bond. With this double bond, it can form, so this double bond can change into a single bond, and with this single bond formation it can form this kind of linear polymers. This kind of linear polymers it can form with the help of this single bond formation. Now what is the role of this bisacrylamide? Therefore this acrylamide can form linear polymers, but bisacrylamide, what it can do is that you can see that bisacrylamide is basically two acrylamide molecules. This is one acrylamide molecule, this is another acrylamide molecule, which is joined by a methylene group. Which are joined together by a methylene group. So that is why it is called methylene bisacrylamide. Now what it does is that it can form branches in the linear polymer of acrylamide. So these branches are formed by bisacrylamide. So therefore in this polymer formation, which is done by acrylamide and bisacrylamide, acrylamide can form linear polymers, and bisacrylamide can form branching in this linear polymer, and therefore thus what it does is that it forms a compact gel matrix. And if suppose if only acrylamide is added and no bisacrylamide is added, then only this polymer will be a linear polymer and it will not form pores, which is needed for the migration of our molecules of interest, be it DNA or protein. Now this acrylamide and bisacrylamide, they form this polyacrylamide gel together, but we have to always remember that we have to mix acrylamide and bisacrylamide in particular ratio. So what is usually done is a 30%, so most commonly done, or I would say that it is a 30% polyacrylamide gel, and where acrylamide and bisacrylamide are mixed in a ratio of 29:1. So here are two factors. One is the gel percentage and another is the acrylamide and bisacrylamide ratio. So by varying these two factors, we can actually increase or decrease the pore size of this polyacrylamide polymer. So how does that happen? So if we increase the gel percentage, then we have to always remember, if we increase the percentage of gel, then the pore size decreases. And so therefore if it is a 12% polyacrylamide gel and if it is a 15% polyacrylamide gel, so then 15% gel will have smaller pores, whereas 12% gel will have bigger pores. So gel percentage, as it increases, the pore size decreases. And the ratio of acrylamide and bisacrylamide is also important. So it can be mixed in several other ratio also rather than 29:1, and that will also determine what will be the pore size or what will be the resolution capacity of this kind of a polymer. So by varying the gel percentage as well as by varying the acrylamide to bisacrylamide ratio, we can make several types of PAGE gels or polyacrylamide gels. Now in making this cross-linked polyacrylamide polymer, there is an important role of two molecules, which are ammonium persulfate and timid. So this is this is a par sulfate molecule, and this is TEMED. TEMED is also known as TEMED is the abbreviation of N prime tetramethyl ethylene diamine, which is in abbreviation called as TEMED. So what is the role of these two molecules, par sulfate or ammonium persulfate and TEMED? This is also known as APS, ammonium persulfate. So this persulfate molecule actually acts as a catalyst. So in this polymerization reaction, so if we mix acrylamide and bisacrylamide together, they do not polymerize spontaneously if we mix acrylamide and bisacrylamide, but there has to be a catalyst that actually helps in polymerization of this acrylamide. So that catalyst is ammonium persulfate. So this has a peroxide bond here, and this peroxide bond can actually generate free radicals. So the role of APS is that it can generate free radicals, but this free radical generation is not enough for polymerization of the polyacrylamide. So what is additionally added in this gel is TEMED, because this APS or the ammonium persulfate can generate free radicals of this TEMED molecule, and then TEMED can initialize the polymerization reaction. So APS is the catalyst and TEMED is the component which initializes this polymerization reaction of polyacrylamide. So this is how the gel can be formed. So there are various other components that are also added in the gel. For example, particular pH buffer, buffer of particular pH, and also other components like this polyacrylamide, which is the ratio of acrylamide and which is a particular ratio of acrylamide and bisacrylamide, APS, TEMED. If it is a native gel, then any denaturing molecules, denaturing agents are not added. But if it is not a native gel, then the denaturing agents, for example like SDS, can be added. So we will discuss later on. But this is the common the common polymerization reaction that takes place while we prepare a polyacrylamide gel by using particular percentage of polyacrylamide along with a particular ratio of acrylamide is to bisacrylamide, and APS acts as a catalyst and TEMED initializes the polymerization reaction. Now in this polyacrylamide gel, there are usually I will be talking more about denaturing gel. By using SDS. So this will be an SDS page, or there can be a non-denaturing gel as well, which is a native page. So in an SDS page, we usually have two types of gel, a stacking gel and a dissolving gel. So what is the requirement of these two types of gel? What we can see here, so this is a common apparatus. These are the glass plates. So these two are the glass plates, among in between which we polymerize the gel. So we place the two glass plates close to each other so that they are approximately, there is 0.5 millimeter thickness in between the two glass plates, and we pour the polyacrylamide mix in between, and let it rest for some time so that the polyacrylamide can form the gel, that is the polymer. So the liquid changes to a solid gel-like structure, gel matrix, and this begins with the addition of APS at 10. Now there are two gel which you can see here, a stacking gel and a running gel or a resolving gel, and on the top of this you can see the wells. So these are the wells. The wells are actually formed by inserting a comb. So comb is inserted to form the wells in this SDS page, and in these wells our samples are actually loaded. So samples are added in this well. Now and then what is done, this sandwich, this glass plate sandwich, it is placed in a tank, in a gel running tank, and we add buffer into it. So this buffer composition is usually tris-glycine. Tris-glycine SDS, or it is called a TGS buffer, tris-glycine SDS. If it is a, if we are talking about a SDS page or denaturing gel, if it is a native page, then we often use tris boric acid EDTA, which is a TBE buffer. So this is for a SDS page, the buffer, running buffer. This is running buffer. This is the composition of running buffer, and this TBE buffer is used for native page. Native means where there is no denaturing conditions used. So then this running buffer is poured in this gel tank, and then there are two electrodes, the negative electrode and positive electrode, and since the proteins, they will be negatively charged. How they will be negatively charged we will discuss, but since they will be negatively charged, they will all move towards the positive electrode. Thus their migration can be visualized after the gel running, that is when we stain the gel. So this is the overall apparatus, gel apparatus. Now coming in more details to SDS page. SDS, this is the molecule, this is actually the molecule, sodium dodecyl sulfate. So what is the role of this molecule, sodium dodecyl sulfate? So what it actually does is that it is you can see that it is a negatively charged molecule, and what it does is that it denatures the protein, and it imparts uniform negative charge to the protein. Now see, our running buffer, the gel running buffer in which we will run the SDS page gel, has a pH of 8.3. Now we know proteins have their specific PIs, which is the isoelectric point. Now if the pH is not same as the PI, or the pH is greater than the PI, or if the pH is less than the PI, then the protein will be either positively charged or negatively charged. Now the protein can be thus positively charged, negatively charged, or if the pH equals to PI, then it can be neutral also. It will act as a Zwitter ion. So now therefore if I have several proteins in a sample, that means there are multiple proteins in one sample, then what will happen, each protein will have their different PI values, and each protein will exist in different forms, either they will be positively charged or negatively charged or neutral in a particular buffer of pH 8.3 per se. So therefore proteins will be differently charged. Now if the proteins are differently charged, they will migrate differently when we apply electric field, right? But we do not want that. So what we want is, we want separation according to the molecular weight of the protein. So that is our goal, separation according to the molecular weight of the protein. So this is what we want to achieve. Now if the proteins are differently charged, then they will migrate differently on the electric field upon application of this electric field. So therefore their migration will be dependent on both its molecular weight as well as its charge. To eliminate this charge factor from its migration, so protein should not migrate based on its charge. So therefore what we have to do is, we have to impart a uniform charge to all proteins, right? So the charge is to mass ratio has to be uniform. So the charge by mass ratio has to be uniform. For all proteins, charge by mass ratio has to be uniform. So to do that, we use SDS to impart this uniform negative charge, or to keep the charge by mass ratio uniform, we use this molecule called SDS. Now you can see in this SDS molecule there is this hydrophobic region here, right? So this hydrophobic region will interact with the hydrophobic region of the protein molecule, and this will actually interact with the hydrophobic region of the protein, and it will impart this negative charge to the hydrophobic region of the protein, to the entire protein. Now how do we prepare our protein sample? So what we do is, we treat our protein samples with SDS, and there is another molecule which is a denaturing agent again, or a reducing agent, which is called beta mercaptoethanol. Beta mercaptoethanol or 2-mercaptoethanol. This is a reducing agent. So when we have a protein sample, we add this SDS and beta mercaptoethanol. So what we actually do is, we add a loading dye. So in a loading dye, we add SDS and beta mercaptoethanol. Now why do we add this reducing agent beta mercaptoethanol? So the proteins, they can have these disulfide linkages. As you know, the disulfide linkages are formed between two cysteine residues. So for example, this, you can see that in this protein chain there can be several disulfide bonds that are formed. Now if these disulfide bonds are not broken before we run the sample on the gel, then this disulfide bond will maintain a particular structure of the protein, and it will not be a linear polypeptide as we want it to be. So therefore that is why to break these disulfide linkages, beta mercaptoethanol or 2-mercaptoethanol is used. So what it does is that it reduces this disulfide linkages, and then what it does is that it makes the polypeptide a linear molecule. So here you can see that the polypeptide molecule is now converted into linear chain, because the disulfide linkages are broken, and because of this SDS is imparting this negative charge. So this, because of this SDS, the SDS molecules are uniformly interacting with the protein and providing an uniform charge by mass ratio to the protein molecules. Now these protein molecules are now a linear polypeptide. You can see that it has been converted into a linear polypeptide. And so to denature the protein samples in this way, we also, what we do is that we heat the sample at 100 degree centigrade, or boil the sample at 100 degree centigrade. So we take the protein sample at these loading dyes SDS and beta barcamptoethanol and boil it in 100 degree centigrade or 95 to 100 degree centigrade. So at this temperature, what happens, the protein first loses its structure, so it becomes linear. Then the disulfide bonds are also broken by using beta barcamptoethanol, and the SDS imparts uniform negative charge on this linear polypeptide molecule. Now they are ready to be run on this gel. So now what will happen, the proteins have uniform charge by mass ratio. Now therefore their migration will only depend on their molecular weight. That means their migration will only depend on how much friction they are going through. They are actually experiencing how much friction, and of course this friction will depend on the size of the protein or the molecular weight of the protein. So the more the molecular weight, the higher will be the friction, and therefore the lesser will be the migration. So here we can say that the larger molecular weight migrates slower, and smaller molecular weight migrates faster. So here we can see that, if I just zoom in this particular gel image, it's a representative image. Here we can see there are 3 samples, and these 2 protein bands, they have migrated more than this particular band. So if we name them 1, 2 and 3, we can see that 2 and 3 have migrated more than 1. So therefore from this we can state that 2 and 3, therefore they will be of smaller molecular weight. So that is why they are migrating faster, their migration is more, and 1 is of larger molecular weight than 2 and 3, that is why its migration is slow. So we can say that the migration is solely dependent on the molecular weight of the protein, and that is how we are able to resolve the protein by according to its molecular weight, so that the small molecular weights moves faster and migrates more in the gel, and the larger molecular weights migrates slowly in the gel because of the friction that they are facing. Now this is the SDS page, and in this SDS page I already mentioned that there are these 2 gel components. One is a stacking gel and another is a separating gel or dissolving gel, and you can see that they have different pH values. So stacking gel has a pH of 6.8. So stacking gel is made with buffer of pH 6.8, and separating gel has a pH value of 8.8. So what is this buffer here? The buffer is tris HCl. So the buffer is tris HCl, and stacking gel has a pH 6.8, whereas separating gel has a pH of 8.8. also another thing that we can see here is that this running buffer has pH 8.3. So why these three different pH values? And what is the function of this stacking gel and this resolving gel? Why these two gel components are needed and why are they made separately with separate pH values? So suppose there is no stacking gel and there is only resolving gel. So by the name resolving gel, we can have an idea that this works as a molecular sieve. The resolving gel it works as a molecular sieve. The resolving gel works as a molecular sieve, which means that it has larger pore size and smaller. It has its particular pore size, right? Now the molecules which are large, they will not be able to migrate through small pores, right? So this dissolving gel, it has this larger pore size and smaller pore size both. Now the large molecules will only be able to migrate through the large pores, but it will not be able to migrate through the small pores. And whereas the smaller molecules, they will be able to migrate through both smaller pores and larger pores. So therefore the smaller molecules, the smaller means the molecular weight small, so smaller molecules will migrate more in the gel. So this is the function of the resolving gel. So it helps in resolving the protein sample. So if suppose there are 3-4 proteins together in one sample, then and they have all different molecular weights, then they will be separated on the dissolving gel according to their molecular weight. But what is the function of this stacking gel then? So by the name stacking, you may have an idea that it actually stacks the protein samples. Stacking means what? So basically, suppose there is a race. You can imagine this: if there is a race and 10 athletes are participating in the race, so then what will happen? You need to start the race from a single line. From one particular line, you need to start the race so that you can actually have an idea that who is going how far and in what time, right? So to understand like how far one athlete has gone, you have to begin the race from a single line, and that is the function of the stacking gel. So what it does is that it stacks the protein samples to a very tight narrow band between the stacking gel and the separating gel or the resolving gel. And how is it achieved? So I already mentioned that this running buffer contains tris glycine. So now glycine, this is the structure of glycine. Glycine has a pI of 6.5. So in our stacking gel, the glycine, the pH is 6.8 in our stacking gel. So in our stacking gel, glycine will be either zwitterion or glycine will be negatively charged, slightly negatively charged, because you can see that the pH is not very not much more than the pI of the glycine. The pH is slightly more than the pI of the glycine. So therefore the negatively charged population of glycine will be in very small amount. So the mostly glycine will remain as a zwitterion in this particular stacking gel. And in pH 8.8, which is much higher than the pI of the glycine, then glycine will be totally negatively charged. Now the pI of glycine is 6.5. So I am assuming that glycine remains either in zwitterion forms or slightly negatively charged. Now in this buffer component there is HCl, so there are a lot of chloride ions because the buffer is tris HCl, so there are chloride ions. Now chloride ions are very small ions, so obviously when we apply an electric field, the chloride ions, they will migrate fastest. This is the positive, this is actually the positive electrode, right? So the chloride ions will migrate fastest because they are very small, and they have high electrophoretic mobility. But the glycine ions, if they are sweeter ions or if they are negatively charged, they will migrate very slowly in this stacking gel. And remember that in stacking gel, the acrylamide concentration is also much less. So stacking gel has larger pores than resolving gel. It has larger pores than resolving gel, so the molecular sieve function of resolving gel is not achieved by stacking gel. Rather, these have larger pores, so all the proteins can migrate equally in this stacking gel, more or less equally. Now we can see that the chloride ions, so when we are applying this electric field, we can see these chloride ions are migrating. These chloride ions are migrating the fastest. So they quickly come at the front. So they form this front, chloride front, it is called. So they quickly form this chloride front. Now the glycine is almost neutral in this pH. So there is less glycine ions, because of which there is uniform electric field, right? So there is increase in resistance because there are less ions, so the resistance increases in this portion, in this stacking gel portion. The resistance increases, and as resistance increases, the voltage also increases. Now because of which what happens, now the proteins, now they are ionized and they now start to move. So because of this voltage difference and because of this formation of resistance, the protein ions now start to move. And once the protein ions start to move, then what happens, there is a sandwich that is formed. So the sandwich is formed between the chloride front, which is at the front, and the glycine ions. So between the chloride ions and the glycine, there are proteins. So therefore we can see that it almost forms like a line. So in the bottom there is, or in the bottom there is this chloride ions, and in the top there are these glycine ions, and the protein samples are actually sandwiched between them. So now what happens, these chloride ions, they migrate very fast, and they now come to the junction between the stacking gel and the dissolving gel. And thus what it does is that it also takes the protein molecules on this junction between the stacking gel and the dissolving gel. So now this actually forms a very tight zone or a tight band of the protein molecules. So this forms a stack, or this forms a line from where the proteins now enter the separating gel, and the proteins migrate according to their molecular weight. So if there would not have been a stacking gel, then what would happen? So we in these lanes we add approximately 15 to 20 microliter of our sample. So if we are adding 15 to 20 microliter of our sample, then our proteins are uniform in this liquid volume. And then what will happen, as we keep on adding the samples in these lanes, then some protein molecules will come closer to the line, and some protein molecules will remain at the top. So therefore what will happen, that this stack formation will not happen, and therefore when we now pass the gel, or sorry, when we pass electric field through the gel, then what will happen, the protein molecules will begin to migrate randomly, because of which there will be no precise resolution. So we cannot achieve precise resolution, which is why we use this stacking gel, where there is this pH 6.8, and this 6.8 is maintained so that glycine is slightly negatively charged in this pH, and glycine forms this sandwich. Glycine and the chloride ions forms this sandwich in between which the protein samples are stacked. And now when these glycine molecules, now when it enters the separating gel or the resolving gel at pH 8.8, the glycine is totally negatively charged. The glycine is totally negatively charged. So now we can say that now the glycines also start to move forward, move with much more electrophoretic mobility. So now the chloride ions also move fast, and the glycine ions will also move fast in pH 8.8. And now so they will go in the front, and the proteins, they will now be separated according to their molecular weight, because now in the resolving gel the pore size is smaller, so the larger molecules will face more resistance and their migration will be slower. So that is the function of the stacking gel and the resolving gel. And the running buffer is maintained at pH 8.3, so that the ions can actually create this voltage, or it can actually allow the migration of the gel, because the protein molecules will not be very much charged for this electrophoretic mobility, but the ions, the other ions of this buffer components, they will actually determine the mobility of this gel. Now once the gel is run, so I'll just show you this gel apparatus once. So this is the gel apparatus. So we have our glass plates here. These are the short plates and these are the large plates, and in between the short and large plates we pour our polyacrylamide gel, and we solidify our gel. And these glass plates are stacked, and they are placed between this clamp, and this clamp is placed in this gel cassette, and these are the combs which are used to create the wells, and this is the gel running tank. This is the gel running tank, where so what we do is we take a gel clamp and we place this gel inside the gel clamp, and we put it on this inside this gel cassette, and we place this gel cassette in this whole system in the gel running tank, and we pour our TGS buffer or running buffer, and then these are the leads, the electrodes, the positive and the negative electrodes, and then we connect it to electric supply, and we run uniform voltage or uniform electric field through the gel, so that now the negatively charged protein molecules can migrate to the positively charged anode. Now once this gel running is done, then what we do is we will stain our gel. because once this gel running is done, we have to now visualize our sample, whether there is separation of proteins on the gel or not. To visualize that, we use these staining dyes. So first of all, while the gel is running, we have to ensure that we do not run the gel too much, so that our protein samples can actually get out of the gel, and it is lost. So we should not allow that to happen, which is why we use this particular dye, which is called a bromophenol blue. It is a tracking dye. It is a tracking dye, and it is also negatively charged. So this bromophenol blue is negatively charged, and it has high electrophoretic mobility. So because of which this bromophenol blue is added in the loading dye. So this bromophenol blue is added in the loading dye. So the loading dye contains this SDS beta-mercaptoethanol as well as this bromophenol blue. And what is the role of this bromophenol blue is that when we run the gel, this bromophenol blue migrates fastest. So it migrates below our proteins. So it migrates fastest below our proteins, and by the since bromophenol blue is blue in color, so we will be able to track how much our gel has run. And once the bromophenol blue dye front, once the dye front comes to the bottom of the gel, we should stop the gel electrophoresis so that our protein samples cannot escape the gel. So that's the function of bromophenol blue, but this is only for tracking how much our gel has been run. But for visualizing the protein samples, we use another dye which is called Kumasi Brilliant Blue G250. There are two different types of Kumasi Brilliant Blue. One is G250, another is R250. But for protein gel staining or SDS PAGE gel staining, we use G250 dye. And this G250 dye, it has there is this hydrophobic region here. There are these hydrophilic regions here. So it has both hydrophobic and hydrophilic regions, and with its hydrophobic region, it can interact with the hydrophobic region of the protein, and with its hydrophilic region also it can interact with the hydrophilic region of the protein, right? It can bind to the protein. So now the protein that has been run on the gel, the protein is linear. So it is a linear polypeptide, right? Now the Kumasi blue dye now interacts with the protein and stains the protein. And by the help of this dye, we will be able to visualize this protein sample by our naked eye, and we can also take a photograph of it. So now this is how a gel looks like. This is how a gel looks like after it has been run. So this is a 12% polyacrylamide gel. It is SDS PAGE. So it is denaturing condition. It is run in denaturing condition, and this particular lane, you can see this is a marker. So a protein marker or a protein ladder is what it is. Actually a mixture of known proteins of known molecular weights, a mixture of proteins of known molecular weight, so that when this marker is run on the gel, we can see these bands and we know the molecular weight of these bands. So suppose we know the molecular weights of these bands. Suppose this is 15 kilodalton, this is 25 kilodalton, this is 40 kilodalton, 50 kilodalton, and so on. So we know the molecular weight of these known proteins, and when these protein marker runs on the gel, by the position of this marker band and by the position of our protein bands, we will be able to determine the molecular weight of our protein sample. So now these are our samples: lane 1, 2, 3, 4, 5, 6 and 7. So what we can see here, that this thick band that we can see here, this is actually our protein of interest. So this is our protein of interest, and we can so this is actually a gel which is run after a chromatographic method. So a chromatographic technique has been used, and after which the protein, there was an attempt to purify this protein, and the protein sample was run. After the chromatographic technique, the protein sample is run in this 12% SDS page, and what we can see here is that in these lanes, lane 3, 4, 5, lane 6, we can see that with our protein of interest, first of all, there are several other bands as well, right? So there are other bands also we can see, which we will call as contaminants, because these are not our required protein bands. So these we will call our contaminants. And how do we know that this is our protein of interest? By the molecular weight marker. So here we can see that this is 50 kilodalton. So our protein also runs parallelly with this 50 kilodalton marker band. So therefore we can say that our protein is also of molecular weight approximately 50 kilodalton. So the approximate molecular weight also we can determine, which is 50 kilodalton. And we can also see that there are other bands. So there are contaminant bands here coming. These bands, which are the contaminants here, we can see. And these contaminants, they are not required in our protein. So we can say that our protein is not, after this purification technique, our protein is not 100% pure. And these contaminants that are present, they are of lesser molecular weight than our protein. So therefore we should apply another chromatographic technique possibly to remove these contaminants from our protein sample. So in this by this visualization method, which is the SDS page, we can determine the molecular weight of our protein, and we can also check the purity of our protein by the presence of, if there are presence of other contaminant bands, then we can say that our protein is not very pure. If there are not much contaminant bands, or there are no contaminant bands, we can say that our protein is pure. So these information are also available after we run this gel electrophoresis, page electrophoresis. Now this is all the denaturing page or SDS page. There is also another type of page which is called a native or non-denaturing page. So here the difference between SDS page and native page is that the SDS molecule is not used in this native page, and also beta mercaptoethanol is also not used. So since SDS is not used because it is non-denaturing, so therefore what will happen, the proteins will be of various charges. So it does not use SDS, so the proteins can be positively charged, it can be negatively charged, or the proteins can also remain neutral in a particular buffer pH, right? So now when we load the proteins in these whales, when we load the proteins which are negatively charged, they will migrate towards the cathode. The proteins which are negatively charged, they will migrate towards the anode. So this is the positive anode. So the proteins which are negatively charged, they are migrating towards the anode. Whereas the protein which is positively charged, it will migrate towards the negative electrode, which is the cathode. So therefore both these types of migration we will be able to visualize in a native page. So therefore that is why it is called native, because there is no SDS used, so proteins are not uniformly charged. So here the proteins will not be separated only according to its molecular weight, but the proteins will be separated according to its charge and molecular weight. So both these factors will come into play. The proteins will be separated according to their charge as well as their molecular weight. Now there is an example which I would like to discuss. So suppose this is one antibody molecule, and we know that antibodies are immunoglobulin G. So this is an immunoglobulin G, and we know that antibodies are proteins. Now this is a non-denaturing native page, right? And in this IgG molecule we can see that there are these linkages. These are disulfide linkages. So this is a disulfide linkage, and the two chains of an antibody, this green one, these green ones are heavy chains, and the red ones, they are called light chains. So now these two chains, they are held together by disulfide linkage. Now when we use an SDS page or a denaturing page, then we use this molecule called beta mercaptoethanol, which reduces the disulfide linkages, and it breaks down the disulfide linkages. So therefore if we run this IgG molecule on a native page versus we run this molecule on a SDS page, then what will be the difference in its migration and how many bands we will be able to observe in native page or SDS page. So let's have a look at that. In a native page, the disulfide linkages will not be broken. So therefore this IgG molecule will migrate as a whole. So there will be no denaturation of this IgG molecule, and therefore this molecule will form only one band. So suppose this is the gel, and this can be the molecular weight markers, and IgG will form only one band. Suppose IgG will form only one band because the disulfide linkages will not be broken in native phase, whereas in a denaturing page, these um, disulfide linkages will be broken down. So therefore, in a SDS page, we will be able to see two bands. One is of heavy chain, another will be of light chain. So there will be two bands. One will be of heavy chain and another will be of light chain. Now here there are two heavy chains and two light chains. So these two chains will together form one band. So that is why we will be able to see only two bands. One is that is of heavy chain, another that is of light chain. So this is the difference between a native page and a SDS page or a denaturing page, where the protein molecule will be denatured and the protein molecule will also migrate according to its charge and molecular weight. Now, in addition to this native page, there is another type of page, which is this 2D gel electrophoresis. So 2D gel electrophoresis is basically when there are two dimensions in this. The first one is PI and the second one is molecular weight. So there are two factors for resolution of the protein samples here. The first factor or the first dimension is the PI of the protein or the charge of the protein. So here this is called isoelectric focusing. This is called isoelectric focusing. So where we have a buffer or a column of pH gradient. So from pH 2 to 12 this gradient is formed, and when we run the proteins in this gradient, in this pH gradient, which is the first dimension, then what happens, the proteins will move according to its PI. So suppose a protein has a PI of 5. So what it will do is when it reaches the pH gradient where pH is 5, in that pH gradient the protein will not be able to move any further because it will not have any charge. So it will be neutral in its PI. So then the protein will form a band in this zone. So at pH 5 the protein will not be able to move any further. So that is how the proteins, all the proteins will be separated according to their PI. So if protein A has a PI of 3, it will form a band near the 3 pH gradient, and if a protein has PI 12, it will form a band near the 12 pH. So that is how the first dimension is achieved, that is by isoelectric focusing. The proteins are moving according to their PI. Now in a second dimension, what is done is that a SDS page is done. So so proteins now move according to their molecular weight. So now the proteins are all denatured and then they are run according to their molecular weight. So this is the first dimension, the isoelectric focusing, and this is the second dimension. Now so all the proteins which have moved to, suppose, pH 5. Now many proteins will have this PI of 5 which will move to pH 5 and they will stop there, they will not migrate any further. Now this, when we apply, when we run this proteins which have come to pH 5, now when we run these proteins on gel, then on SDS page, then all these proteins which have come to pH 5 will now be separated. You can see that they will be now separated according to their molecular weight. So these means there are two dimensions of resolutions here applied. So which is why it is called a 2D gel electrophoresis. Now that is all about our polyacrylamide gel electrophoresis. Now coming to another type of gel electrophoresis, which is agarose gel electrophoresis. Now agarose is a linear polysaccharide. This is how an agarose looks like. It is a linear polysaccharide, and what is the difference between an agarose gel electrophoresis and a polyacrylamide gel electrophoresis? So agarose gel has larger pore size than polyacrylamide gel, and the pore size of agarose gel can be controlled only by the percentage of agarose. So suppose we have 1% agarose gel and 0.5% agarose gel. So 1% agarose gel will have smaller pore size, and 0.5% agarose gel will have larger pore size. So the pore size of agarose gel can only be controlled by the percentage of agarose. Whereas we had seen that in case of polyacrylamide gel, the pore size of polyacrylamide gel can be controlled by two factors. That is, one is the acrylamide percentage, and another is the ratio of acrylamide to bisacrylamide. So therefore the pore size of polyacrylamide gel can be controlled more finely, which is why the pore size of polyacrylamide gel is even lesser than agarose gel. So if we have bigger molecules, then we run it on agarose gel. So for example, DNA is preferentially run in agarose gel if the DNA molecule is suppose higher than approximately 200 base pairs. So then from 200 base pair to approximately 10 to 20 kilobase pairs, this huge range of DNA samples can be run and resolved on agarose gel electrophoresis. Now this is how the agarose gel setup looks like, and here we can see that this is actually a typical agarose gel and similar to polyacrylamide gel. But remember that in case of an agarose gel electrophoresis, this electrophoresis is run horizontally. So this is a horizontal setup. And whereas we had seen in this setup that this was actually the gel cassette and the gel was run vertically in case of a polyacrylamide gel, but agarose gel is usually run horizontally. And here also there are two electrodes, a positive electrode and negative electrode, and we know that DNA and RNA, they have this phosphate backbone which is negatively charged. So these two molecules are negatively charged. So therefore when we apply voltage, so these molecules will migrate towards the cathode. They will migrate towards the anode, which is the positively charged electrode. So they will migrate towards the anode. Now how does this polymerization of agarose occur? So these agarose molecules, they initially form this helix kind of structures, but when we heat them approximately at 100 degrees centigrade, then they form this fine gel structure, and these are the pores that form between these agarose polymers. Now the agarose gel percentage and the DNA resolution, um, it can be controlled very carefully. And so suppose we can see here, this is a chart of the agarose gel percent and the DNA size resolution, and we can see that if the agarose gel has a percentage of 0.5%, then it can resolve approximately from 1 kilo base pair to 30 kilo base pairs of DNA. If it has a higher percentage, then what will happen? The pore size will decrease, and if the pore size decreases, then it will not be able to resolve larger molecules. So now it will be able to resolve smaller molecules. So from 0.5% to, if we go to 1.5%, for example, we can see that in 1.5% we can resolve from 200 base pair to 3 kilo bases of DNA, right? So from this we can see that as we increase the agarose gel percentage, our resolution also increases. That means we can now resolve smaller DNA molecules as the pore size is getting smaller as we are increasing the gel percentage. So in case of 0.5%, the pore size is small, and in case of a 2%, the pore size is smaller. So in case of 0.5%, the pore size is large, and in case of a 2% gel, the pore size is actually smaller. Now if we see how do we cast an agarose gel in our lab, so what we do is that, first of all, what is the buffer in which we run agarose gel? The buffer is TAE, which is tris acetic acid and EDTA. Tris acetic acid and EDTA. Why is EDTA used? Because EDTA is a chelating agent, and EDTA can chelate the ions, the magnesium ions, for example, and or zinc ions, for example, and it does not allow any nuclease or protease to function. So that is why EDTA is added in this TAE buffer. Now what we do is we measure a particular volume of this TAE buffer, and we add powdered agarose to it. So suppose we are making a 1% agarose gel, and the volume of the final volume is 50 ml. 50 ml of 1% agarose gel we are making. Then since it is 1%, then how much agarose powder do we need to have? So we need to have 0.5 gram of agarose powder. So this is basically 1% weight by volume. 1% weight by volume. So therefore we will use 0.5, we will actually weigh 0.5 grams of agarose powder, and we will mix it with the TAE buffer. Then we will heat the solution. Now once we heat the solution, now this agarose powder will actually melt, and it will form a liquid. So we mix the liquid properly, we swirl it to mix the liquid, and then we add a stain or a dye. So this dye is usually ethidium bromide. We have to remember that ethidium bromide is an intercalating agent. So it intercalates between the bases of the DNA. This ethidium bromide is an intercalating agent, and so basically it can fluoresce when we observe it under UV light, and that is how we can visualize our DNA after our gel is run. So now that we add this DNA stain or ethidium bromide, or it can be any other types of stain also, and then we mix the stain with the in the liquid, which is the gel, and now we pour the gel on this casting tray. This is the gel casting tray, and we pour the gel, and we use the combs here to form the wells. And on this casting tray, if we leave the gel for approximately 30 minutes, then what we will see is that it will solidify and it will form a thick gel. So the difference between a polyacrylamide gel and agarose gel is that a polyacrylamide gel is only 0.5 millimeter thick, but for example agarose gel can be approximately 1 centimeter thick. So it can be from 0.5 centimeter to 1 centimeter thick also. So it's a much thicker gel, and its pore size is also much more than a polyacrylamide gel. So this is how we are casting the agarose gel, and then we subject the gel to electric voltage. So then we run the gel, and these are our wells where we load the samples. So we now keep the gel on a gel running tray or a gel running tank. We dip the gel on a gel inside a gel running tank in this gel running buffer, which is again TAE, and we add our samples in these wells. Now when we add our samples, we also have to add this DNA ladder. So this is similar to the protein ladder also. The DNA ladder contains specific DNA sequences or specific DNA molecules of known molecular weight, and as they migrate on the gel, we can assume or we can have an idea of the molecular weight of our samples as well. Now this is the DNA ladder. We run the DNA ladder on the gel, and on other wells we add our protein samples. Now when we are adding our protein samples, we also add this dye similar to our SDS page. We also add this bromophenol blue here to track the migration of the gel. So this is the tracking dye again. And so we place the gel in such a way that we are placing our samples away from the positive electrode, so that our samples can move towards so this is the direction of the movement. So our samples can run towards the positive electrode. Our samples can run towards the positive electrode. So we place the samples closer to the negative electrode, so that the samples can now run towards the positive electrode on application of the electric current. And remember, if we place the gel in an opposite direction, that is, if we place the sample towards the positive electrode, then what will happen? The samples will move out of the gel. So then we will not be able to visualize our sample. So we have to maintain this direction, so that the negatively charged DNA molecule can all move towards a positively charged electrode. Now, how is this migration on the agarose gel resolved? So basically, the DNA molecules which have higher size or which have larger size will migrate slower, similar to polyacrylamide gel, and the DNA molecules which are shorter or which have small size, they will migrate faster. So the shorter DNA fragments will come down in the gel, whereas the longer DNA fragments will remain on the upper part of the gel, because they will face more resistance. So their migration will be slower. And this is actually a gel that we have run in our lab, and This is again the DNA ladder, and their molecular weights are known. So for example, from the top it is a 10 kb, then the next band is 8 kb, 6 kb, 5 kb, 4 kb, 3.5 kb, 3 kb, 2.5 kb, 2 kb, and all that, right? So these are the known molecular weights of these DNA fragments, and we can see that we have run our sample in lane 1 and lane 2, and in lane 1 we can see a DNA fragment which is close to the 5 kb marker band, and in lane 2 we can see a DNA fragment which is close to the 2 kb marker band, right? Now therefore we can more or less have an idea of this molecular weight of these DNA samples. So this one, since its molecular weight is approximately between 2 to 2.5 kb, so therefore it's what to say, it is a smaller DNA molecule, so which is why it has migrated farther in the gel, whereas this has a larger size which is approximately 5 kilobases, so which is why it has migrated less in the gel. So from this migration we can have a comparison of the DNA sizes. We can have an idea of the approximate size of the DNA, approximate molecular weight of the DNA, or approximately what is the how many bases are in this DNA. That idea we can get after we resolved our DNA samples on Agarose gel. So that's all about gel electrophoresis. Now let's see the questions based on gel electrophoresis. So the first question is, in gel electrophoresis the gel acts as molecular glue, molecular copy machine, molecular sieve, or molecular scissor. The correct answer is the gel acts as a molecular sieve. Second question, which is a suitable technique for separating large DNA fragments? The correct answer here will be Agarose Gel Electrophoresis. Since the pore size is larger on Agarose Gel Electrophoresis, so it is recommended for separating large DNA fragments. Next question. Which of the following can be determined by using SDS PAGE? The correct answer is the molecular weight of the protein as well as presence of disulfide bonds in a protein. So if we run the protein sample in SDS PAGE, it will reduce the disulfide bonds, and therefore we will be able, so if there are two chains of a protein which are linked by disulfide linkages and they are of different molecular weight, we will be able to see the two chains on the SDS PAGE, which is very similar to what we just had shown for the IgG molecule. Next question, what is the role of SDS in SDS PAGE? So the correct answer is it imparts uniform negative charge to the proteins. Next question, what is agarose? Protein, polysaccharide, DNA, or lipid? So agarose is a linear polysaccharide. That is the correct answer. Next question. Migration of a protein in SDS page is determined by which of the following? Charge, size, shape and PI of the protein. Which of the following determine the migration of protein in an SDS page? So the correct answer is only the size of the protein. The molecular weight or the size of the protein determines the migration. Next question. What is the advantage of using separate gels for stacking and resolving? So the correct answer here is to increase the resolution of separation, because the proteins are now stacked on a single line. So now it will be easier for them to resolve them. It will be also precise when they will be dissolved in the separating gel or the resolving gel. So the other options like the increase the migration rate or maintain the charge of the protein or retain the shape of the protein, these are not correct. So only the correct answer is the resolution of, to increase the resolution of separation. Next question. What is the role of APS, ammonium persulfate, in SDS page or native page? So the correct answer is to provide free radicals for cross-linking reactions. So it acts as a catalyst to provide free radicals, so that now the TEMID can cross-link between the acrylamide and bisacrylamide. Next question. What are the major ions present in the buffer in SDS page? So the correct answer is all of the above. All these ions, tris, glycine, chloride, all these ions are present in the buffer. Next question. Beta mercaptan ethanol or BME is used to break the hydrogen bonds, break the disulfide bonds, break the peptide bonds, or break the multimeric proteins into monomers. So the correct answer here is that beta ME breaks the disulfide bonds. So it is a reducing agent. Next question. What is a non-essential component of an SDS page? Acrylamide, tetramethyl ethylene diamine, ammonium persulfate, or beta mercaptan ethanol. So now in an SDS page, only the beta mercaptan ethanol can be cannot be used, but other materials like the acrylamide, TEMID, APS, these are all required for formation of the gel polymer, but beta mercaptan ethanol cannot be used. So it's a non-essential component. If beta mercaptan ethanol is used, then it will be a denaturing page, and there will be it will be acting as a reducing agent. Next question. Which is the staining method for protein gels? The correct answer is massive brilliant blue staining. Next question. How are proteins separated in a native page? The proteins can be separated based on all these factors, that is size, charge and PI. Because there is no SDS, so proteins have different charges and their different PI values are determining where the proteins will be migrating, and also the size of the protein, which is the molecular weight of the protein, which will determine how much friction the protein molecules will face. Next question. The pore size of the polyacrylamide gel can be changed by changing which of the following? So the correct answer is all of the above, because I already told that by changing the concentration of acrylamide and bisacrylamide as well as the ratio of the acrylamide and bisacrylamide, we can change the pore size of the polyacrylamide gel. Next question. In electrophoresis, DNA will migrate towards which electrode? Now DNA is negative, so it will migrate towards positive electrode. What is the positive electrode? The positive electrode is anode. So DNA will migrate towards the positive electrode and not the negative electrode, which is the cathode. So the correct answer is anode. Next question. What is to be done to reduce the pore size of agarose gel? To reduce the pore size, we have to increase the agarose percentage. How can I visualize DNA in gel? The dye which is used is ethidium bromide, which intercalates in the bases of the DNA. On what factors this migration of DNA depends on the agarose gel? So the correct answer is shape and size of the DNA molecule. So DNA shape is also important. So suppose the DNA which is single stranded, it will always migrate faster than double stranded DNA. So single strand DNA migrate faster than double strand DNA. Suppose if it is a linear DNA and it is a supercoiled DNA. So plasmid DNA, with which we usually work, we clone in a plasmid DNA. So these kind of plasmid DNAs always are present in supercoiled form. So if the plasmid DNA is circular, it exists in a supercoiled form. So supercoiled DNA will again move faster than linear DNA. So supercoiled DNA will actually form band lower than the linear DNA. So both the shape and the size, which means the bases, how many bases are present in the DNA, those two factors will determine the migration of DNA. Which of the following is true for SDS PAGE, native PAGE and agarose gel? So the correct answer is A, B and D. In SDS PAGE, all proteins are negatively charged, that is correct. In native page, proteins can be negatively charged, positively charged, or neutral. That is also correct. And in no denaturing agent is added in agarose gel electrophoresis. That is also correct. But this one is incorrect, that nucleic acids are positively charged. Nucleic acids have their inherent negative charge. So that is an incorrect statement. So the solution is A, B and D. And the last question for today. This also has multiple answers. Okay, so correct answer is that agarose gel can be depolymerized by heating, but a polyacrylamide gel cannot be depolymerized by heating. In the absence of bisacrylamide, acrylamide forms a linear polymer. That is also correct. Bisacrylamide acts as the cross-linking agent. And the another correct option is that when it is easier to control pore size in polyacrylamide gel than in agarose gel, because in polyacrylamide gel there are two factors. One is concentration of the acrylamide or percentage of the acrylamide, as well as the ratio of the acrylamide and bisacrylamide. So these two factors can be controlled much more easily, and that is why the pore size can be controlled in a much better way than agarose gels. So that's all for today, and thank you for listening to us. And this is the 6th week of live session of experimental biochemistry. We will meet again on the next week, same time, Tuesday, 6 to 8 p.m. Thank you.